Welcome, everyone. Thank you so much for joining us this afternoon. If you don't know me, my name is Rachel Schrader. I am Vice President of Clinical Care and Education here at PPMD, and I am thrilled to be joined by our colleagues at REGENXBIO today for an update to the community on their AFFINITY DUCHENNE pivotal trial and some interim functional data. I know that our speaker, Naz, has been really busy this week sharing some updates with the community at a few different venues. And this afternoon, we get to share this with parents and caregivers and everyone who wants to tune in. So we're really excited to hear about some of this really exciting interim data. We are recording today's webinar. So if you want to go back and listen or share with a friend, you'll be able to do that after the conclusion. And we are taking questions. So if you have questions for Naz or for Olivier, please feel free to drop those in the chat. And after the conclusion of the presentation, we'll go to an open Q&A. So with that, I would love to introduce our colleagues that have joined us today from REGENX. We have Dr. Naz Dastgir, who is the Senior Clinical Development Lead and will be giving us the overview presentation today. We're also joined by Olivier Danos, who is the Chief Scientific Officer and will be available to answer additional questions during that Q&A session later on as well. So with that, I don't want to take any more time than I already have. Naz, I'm going to go ahead and hand things over to you. We're so excited to hear about what you have to share with us today.
Thanks, Rachel. So I'll just jump right in so we have more time for questions from all of you joining. So I appreciate all of you for being here today. So we're going to talk about the RGX-202 AFFINITY DUCHENNE pivotal trial and interim functional data. My name is Naz Dastgir. As Rachel mentioned, I'm the clinical development lead on the program here. These are our forward-looking statements. And here's the agenda for today. So I'm going to give you an overview of REGENXBIO, our company, and RGX-202, our Duchenne muscular dystrophy gene therapy. We'll go over the phase 1/2 study design and data. We'll also review the new biomarker data and the first functional data that we have. We'll also talk about our pivotal study, which has opened up. Yay! It's our phase 3 study as a trial for RGX-202, and then go through a question and answer session. So let's talk about RGX-202. So RGX-202 is designed for improved function in Duchenne. RGX-202 is the only microdystrophin gene therapy with the functional elements of the C-terminal domain, which is found in naturally occurring dystrophin. So here's a listing of RGX-202 compared to some other drugs that are either commercially available now or in research. When you deliver gene therapy, we use an adeno-associated virus. These vary between companies. We have chosen AAV8. We also use a promoter called SPC512. The promoter helps the transgene get to the right location, which we primarily focus on being the skeletal muscle. And then you have a specially formulated transgene. This transgene is designed based off of what's been learned in prior studies on some Becker patients that were very high functioning. And some really smart scientists got together and they said, what is unique about these Becker patients that are really high functioning? What are the components of the gene that are essential to providing function? Because that dystrophin gene is like a little too big to package into these tiny vectors. So each company selects different components of the gene, packages it in, and we uniquely have the addition of a C-terminal domain. So you see that CT domain. So I'm going to talk to you about the role of the CT domain. So it helps with preserving muscle health. So preclinical studies in mice indicated that the CT domain in RGX-202 microdystrophin enables muscle resilience. So if you look at the figure here on the left side, you can see that there's on the y-axis here, this one going up, there's a force measurement. And in a control mouse, a mouse without Duchenne, we can see that the force is around, just about 70. In a Duchenne mouse, the MDX mouse, we see it drops to about 20%. Well, we tested the Duchenne mouse with our microdystrophin without the C-terminal domain, and we got to around 40%. But then when we added the C-terminal domain within our microdystrophin, we got even more, so closer to the 60%. So that's why we made the decision to include this component and kind of make it a better version of what we think it could be just with the microdystrophin without the C-terminal domain. So how it may contribute to improved function. So the CT the CT domain, the C-terminal domain, significantly enhances restoration of the DAPC. This is the dystrophin-associated protein complex. This is a complex of proteins that exists in the muscle membrane, kind of like scaffolding. So this helps with the restoration of this DAPC in the MDX mouse, and it's more similar to natural dystrophin. There's also prolonged microdystrophin activity. So the CT domain increases the half-life of RGX-202, which is the half-life means how long it takes to get rid of about 50% of the drug, which remains in the muscle fibers longer and then helps kind of strengthen the muscle fibers even better. And in terms of muscle health, the CT domain also in the RGX-202 microdystrophin also protects against contraction-induced damage to enable better muscle recovery. So we're hoping to get a lot of benefit from this CT domain. So I'm going to go into the phase 1/2 study that we have. So the phase 1/2 study is the study that we initiated, and we still have going on in some sites that haven't yet rolled over into our pivotal protocol. But it'll hopefully be fully enrolled soon. So I'll give you data on, which areas of the phase 1/2 study are still enrolling. But obviously we have other sites where the pivotal study is up and running already as well. So the phase 1/2 AFFINITY DUCHENNE study, so phase 1/2 study's primary goal is to check for safety and tolerability of the drug, okay? So in this study, we started off with dosing boys who were 4 to 12 years of age. And we started off with our lowest effective dose, which is dose level 1 times 10 to the 14th genome copies per kilogram. We dosed 3 boys at dose level 1 and got an okay from an external safety committee meeting to move on to our next dose that we were evaluating, which is dose level 2. Dose level 2 is 2 times 10 to the 14th genome copies per kilogram. Once we dosed 2 children safely with that, see that green check mark, we checked that off. We then got the okay to move on to a dose expanded cohort of dosing 5 additional patients with 2 times 10 to the 14th. And these 5 additional patients were 4 to 12 years of age. But then at that time, we also said, you know what, there's an unmet need. Now with newborn screening coming out, with families learning about genetic diagnoses in utero, let's go ahead and open up another cohort of patients that we can dose at 1 to 3 years of age. Obviously, it's going to be hard to see the benefits of the drug. This is a new novel area that we're going into in these younger kids, because sometimes the unique feature of Duchenne is that it might take a while naturally, if you didn't know about the genetics, to kind of display your manifestation of Duchenne muscular dystrophy. But we hope to be able to assist with some of the developmental delays that the kids might have, and also show over time that the drug is durable and sustainable and can actually benefit these kids, and maybe even benefit them even better by giving it at a younger age. We don't know. We're still learning more about this younger age group. So as a result, in the study, we opened it up to 1 to 11 years old at screening. At this time, the kids need to have genetically confirmed Duchenne muscular dystrophy. So this is the mutation exemption right now is that at present, we are only allowing children with deletions and duplications in exons 18 and above. But we are working very actively to change this, hopefully in the very near future, so that we can expand our inclusion criteria to allow for more patients to be included. So please keep your eyes open, and obviously we'll inform PPMD if and when this comes to fruition. In order to enter the study, you should also be able to complete a 100-meter walk if you're 4 to 12 years of age, or complete a 10-meter walk if you are 1 to 3 years of age. And of course, at this time, no pre-existing antibodies to AAV. So we don't have a titer cutoff. You're either positive or you're negative. And so if you are positive for the AAV8 capsid at this time, we unfortunately cannot allow you to participate in the study, not because we don't want to, we would love to, but the issue is that it would potentially render the drug ineffective, and also make it potentially a worse immune response to the drug because of your prior existence of antibodies. So in the box below, I'm going to go through the administration assessments and timelines. So in the phase 1/2 portion of our study, we had some baseline assessments that needed to be done so we could see how the kiddos were at baseline and how things changed over time after dosing with RGX-202. So at baseline, we did get a muscle biopsy, which we're then following up with a muscle biopsy 3 months after dosing. This is to make sure that our transgene gets into the right place in the muscle tissue and starts producing the microdystrophin protein. We can also do something called immunofluorescence, which we'll show on a subsequent slide, that will allow you to show that the protein is being picked up in the rim of the muscle cell or the sarcolemma, the muscle membrane, and localizing to the right place. So not only are you producing the protein, but you're also localizing it in the correct location. So, these are the two muscle biopsies we do in the study. We don't do additional muscle biopsies. We also do muscle strength and functional assessments and muscle imaging prior to dosing. And then because of some concerns about serious adverse events in other studies, as we were designing our study, we did our best to try to do our best by the families and come up with an immune suppression regimen that would prevent any serious adverse events, which obviously we've seen in prior studies, which no one wants to ever be a part of. Obviously, every company is in this to help children, and so it's something that we all fear and we all work collaboratively as a group to prevent. But we made the unique decision to start a little bit more of a comprehensive immune suppression regimen, which involves 3 different drugs that we start before dosing. We continue for about 2 months after dosing and then taper down in the 3rd month. And then that's it. You have about 3 months and a week or a week and a half of immune suppression that you're on, and then you're good to go. So I think it's a really good investment. When I go through our safety profile, you'll see why. And then subsequent to the dosing, we continue to do muscle strength and functional assessments and additional muscle MRI studies. So here are some key baseline demographics of the patients that we enrolled in the study. This is a lot of data on this slide. I just put it here. We don't have to go through the nitty-gritty detail here, but uniquely, we did dose older children in this study. So we have about 7 kids who are 4 to 11 years of age with dose level 2 that were dosed. We've gone up in dosing to 35.5 kilograms, which is higher than some other studies that are offering access to gene therapy right now. We've also had a bit of a more permissive time to stand requirement. So we're allowing less than 9 seconds for entry into our study. We actually had no one enter who was greater than 7 seconds. So we actually altered that criteria for our study. So for the pivotal study, so you just need a time to stand of less than 7 seconds to be able to enter into the study. And obviously to be able to complete the 10-meter walk run and the 100-meter walk as well. So you can see that we have dosed kids with a time to stand of up to 6.8 seconds with dose level 1, up to 5.4 seconds with dose level 2. And this is kind of where we're at with our baseline characteristics of our patients. And in these patients, thank goodness, cautiously optimistic, always knocking on wood, we have not had any serious adverse events, any adverse events of special interest, any myocarditis, or any myositis. The most common adverse event that we have had has been nausea, vomiting, or fatigue, which have all resolved. So we find that RGX-202, along with our immune suppression, has been well tolerated in all patients at both dose levels with no serious adverse events or adverse events of special interest. So this is a great source of pride for us. Obviously, we know that sometimes certain things are inevitable, but we do our best as a company to put this priority as priority number 1 as we continue to explore the functional potential of RGX-202. So let's talk about some of the biomarkers. I know this is heavy on the science, but I feel like some of you in the crowd might be interested in seeing this. So biomarkers support consistent, robust expression, which is showing that the microdystrophin is actually being produced, transduction, which shows that the virus is getting into the tissue, and sarcolemmal localization, meaning that the protein that's being produced isn't just sitting in the muscle cell, it's actually localizing on the muscle membrane where we need that scaffolding support. So we have our data here listed for both dose level 1 and then dose level 2. Obviously, there's a lot of data here, but I'll focus on dose level 2 because that's our goal dose for the study. That's what's going to be administered in the pivotal study. So far at dose level 2, we've dosed 1 child who was 4 to 7 years of age and 5 children who were 8 to 11 years of age that have biopsy data. As I mentioned before, we've dosed 7, but we haven't hit the 3-month mark for 2 of them. In the 4 to 7-year-old, the microdystrophin that we were able to quantify on a study known as a Western blot was 77.2%. In the 8 to 11-year-olds, it was an average of 39.7% with a low of 20.8 to 75.7 as a high. Vector copy per nucleus is the amount of copies of the vector, the viral vector, that gets into the cells themselves. So this is listed here. We have 55.4 copies per nucleus in the 4 to 7-year-old age and 17.8 in the 8 to 11-year-old range. And immunofluorescence is shown here on the right side. So this is more of a fiber intensity picture, which we thought was very striking and pretty to look at, which we'll show you in a minute. But typically when you look at immunofluorescence, you're also looking at the rimming of the fiber. So if there is rimming of the fiber, the fiber is colored in, and the fiber are all these polygonal shapes of the muscle tissue here. And what we're seeing in the picture on the right shows that in those fibers that have the rimming, which is anything that's not blue, so the blue fibers don't have the rimming, but anything that's not blue has rimming, that those that are rimmed have different levels of intensity. So there's either low, moderate, medium, or strong intensity that we see. So the majority are turning into the low to medium intensity, but you'll see some high intensity. And this is only at 12 weeks. So we are always encouraged to show our data earliest signs as quickly as possible, but we anticipate, especially with our drug, that if we were to check this muscle biopsy, which we would love to do, but we wouldn't do because we don't want to be too invasive by repeating biopsies on children. But if we were to check a muscle biopsy in 12 months, I wouldn't be surprised if all of this turns like a red color or something, because it takes time for this to deposit in here. But unfortunately, we can't speak to that at that moment. We can only speak to what we see right now, which after 3 months, we're seeing really good deposition of microdystrophin in the sarcolemma or in the muscle membrane. We know that our drug is getting to the right place. We're knowing that a good amount of drug is already being produced and able to be quantified at 3 months or 12 weeks. So this is all very reassuring. So now I'm going to talk to you about our functional data. So what we're going to present here is obviously data from baseline in the children. This is because it's a phase 1/2 study. We don't have a placebo control, and actually we're not anticipating doing a placebo control study for our pivotal study, which I'll go through with you later. But in that regard, regardless of that, what I'm going to present to you right now is our phase 1/2 data. So you'll be able to see how the children perform or improve compared to baseline, but you'll also be able to see how they compare to an external control, because as everyone knows who has a child with Duchenne or treats children with Duchenne, once you hit a certain age, typically around 7-ish, the trajectory is such that you start to see some form of a decline. It's a plateau or a decline, especially depending on whether or not you go on steroids, for example. And at some point, that decline accelerates. And we want to be able to capture how our drug is performing against this decline. So there's been a lot of people working on creating natural history data sets that are available for us to compare our data to as the children age in our study. And we compare our patients by matching our patients to the natural history data set. And the criteria that we use for matching is we'll take one of our children in the study, for example, and we see what their baseline time to stand, time to run, walk, and time to climb are, for example. And then if we have 12 months of data on that patient. And then we check and see if there's someone in the data set, in the natural history data set, that has the same baseline, and then we compare it to how that child is doing 12 months later. And that's how we can compare to the natural history control. So that's how we're going to present the data, but obviously you'll also see the baseline improvement on the charts. So here's our dose level 1. So dose level 1 is our lowest effective dose that we started off with. And just even with dose level 1, our participants demonstrated improvement in function and exceeded external natural history controls at 12 months. So these are the average values of the 3 patients who were dosed with dose level 1. So again, like I said, the improvement over 1 year is listed as an improvement as compared to natural history. But you can also see how the average is improving past baseline. So compared to natural history, what you would expect in a child a year out, we have we've gone down in a time to stand seconds of 2.9 seconds. Our 10-meter walk run improved by 1.1 second, and our time to climb improved by 1.8 seconds. On the far right here, we also have our North Star Ambulatory Assessment Scale grades here. And you could see that the average increase in NSAA score, where your increase is a sign of improvement, and the maximum score is 34. Our patients have improved by at least by 4 points on the NSAA when you compare them to natural history. But you can see just from baseline, on average, our patients who were 4, 6.6, and 10.5 years of age at dosing, they improved by about 2.3, and that's with our lower dose. Then we also wanted to see how our drug was comparing against some benchmarks that have been set by the FDA in the approval of Elevidys in ambulatory boys. So Peter Marks had referenced something from a paper called the Minimally Clinically Important Difference, or the MCID. And that is listed as a velocity. And we're comparing the average velocity of improvement, average improvement in velocity of our patients to the MCID. And you could see that our patients in blue are outperforming the MCID in time to stand, 10-meter walk run, and time to climb velocity at dose level 1. Again, not our selected dose. So then we go to our pivotal dose, which is our higher dose, or dose level 2. So here you can see the data that we have from our patients at 9 months. So we can't compare to the MCID because the MCID is at 12 months of age. So this is the early data that we're able to share, and we hope to get more data and we can share with you later. But this is what we have right now. But you can see that the 8-year-old, compared to natural history, had an improvement of 0.9 seconds. The 10-meter walk run improved by 0.7 seconds, and the time to climb improved by 1.2 seconds. And in our 12-year-olds, this child improved by 3.1 seconds on the time to stand from baseline compared to natural history. He improved by 1.3 seconds on the 10-meter walk run and 2.7 seconds on the time to climb. We don't have a comparator for the 12-year-old on the NSAA here, so there's no little gray box. But even if you just look at his improvement from baseline, his NSAA improved by 10 points after dosing. So pretty remarkable. We're obviously watching how he continues to do, how all of our children continue to do. We anticipate them to hopefully get better and have good durability of the drug over time. But we're very excited by these results. And this is an average change between the two patients that we have. Obviously, it's a small number of patients. These are 2 patients. We need time to kind of demonstrate more efficacy, obviously, but these early signs point to a good trajectory for how our drug is doing, particularly at dose level 2. So our average time to stand improvement compared to natural history was 2 seconds. The average time to 10-meter walk run improvement was 1 second. The average time to climb improvement was 2 seconds. And our average NSAA improvement was 7.3 seconds compared to natural history. In conjunction with the functional assessments that we obtain, we also ask parents to give their own opinions on how they think their children are doing. So these are patient-reported outcome measures, and caregivers have reported improvements in the home and community environments as measured by one of our patient-reported outcome measures known as the POTSI. The improved skills include running, riding a bike or tricycle, climbing stairs, walking in the community, or participating in recreational activities and sports with peers. So in summary, we'll go through here. We've had positive safety, biomarker, and functional data that demonstrates the potential of investigational RGX-202 to be a differentiated best-in-class gene therapy. It's been well tolerated in 11 patients across both dose levels with no serious adverse events or adverse events of special interest. And the biomarkers that we saw support consistent, robust expression, transduction, and sarcolemma localization, which I think you guys all know what that means. So it's getting the drug to the right place in good amounts and localizing in the muscle membrane. And the participants treated with RGX-202 demonstrated clinically meaningful improvement in functional outcomes at both dose levels, with the comparisons exceeding natural history controls and the minimally clinically important difference that we noted that the FDA had mentioned in the approval of ELEVIDYS and even vamorolone. So our pivotal trial is now active and recruiting ambulatory boys ages 1 and over. So there's no age limit in our study. And we plan to use RGX-202 microdystrophin as a surrogate endpoint, likely to predict clinical benefit and support FDA accelerated approval. So I'll go through the phase 3 trial design to kind of explain that a little bit more clearly for you. So accelerated approval is something that you can do if you can use a biomarker to say, our drug's getting to the right place. We anticipate that this is going to be a good marker of clinical improvement over time. But obviously, it's going to take time for us to demonstrate clinical improvement. So just so we can get patients access to the drug as quickly as possible, FDA, will you allow us to use our microdystrophin levels for accelerated approval? So we got alignment with the FDA on that. So we're going to use our microdystrophin levels for accelerated approval. But then, of course, after you get accelerated approval, you then have to go for a traditional approval, which then uses the functional data to kind of support that statement that you make that the microdystrophin that you're producing is enough to indicate that there's likely clinical benefit from the drug. So we hope as a company to file for a biologic license application in 2026 and use this accelerated approval by after we get data from 30 patients who have received dose level 2. And the eligibility criteria for our study, which is our pivotal study, is boys ages 1 and over who are weighing 10 kilograms or more. The boys do need to be ambulatory at this time, just so we can get some outcome measures from them, just to kind of try to indicate some level of functional benefit. So just please note that they need to have genetically confirmed Duchenne involving deletions or duplications in exons 18 and higher. But like I said, keep your eyes peeled for potential modifications to that. And you must also have no pre-existing antibodies. Our primary endpoint is going to be the proportion of patients with greater than 10% microdystrophin levels. And our secondary endpoints are primarily our functional endpoints, which include the time function test, which we reviewed today, the time to rise, the time to 10-meter walk-run test, and the time to climb. We're also going to institute the stride velocity 95th centile, which is a wearable device to try to see if we can capture changes over time, particularly in the 1 to 3-year-old age group, and we're using it as an exploratory endpoint in our 4 and over older patients. And as an exploratory endpoint in our older patients, we're also checking the North Star Ambulatory Assessment. We have a few active and recruiting sites right now. We have Lurie Children's in Chicago, Virginia Commonwealth University Children's Hospital in Richmond, Arkansas Children's Hospital in Little Rock, UT Southwestern in Dallas, Texas, and Stanford University at Palo Alto. So we hope to open up more to get you to have easier access. I'll tell you here that we support travel. So if you're interested in being in our study, we do support travel to get to the site. We want to try to make the experience as easy as possible for you. And obviously, over time, we hope to have more dots on the map here for you, so you have closer sites for you to be able to be enrolled in the study. I just wanted to put out a huge shout out to our team. First of all, team includes the participants in our study. Even though I don't get the benefit of seeing their beautiful faces in my current role, we are so indebted to the families and the patients for being a part of the study. Obviously, they took a risk on us. I hope they're feeling like they have good benefit. And they're really changing the history of the field by being a part of the study. So we are forever as a company indebted to them and very grateful to them. And additionally, we had the support and help of wonderful PIs at the different sites listed here. And of course, things would never happen without their study coordinators. And in the end, please know that behind all of this is a very loving team who cares about this population of patients so very much. We're working tirelessly for you. Everybody on here is very dedicated and very excited to see what we could bring to the table for the Duchenne community. If you have any questions, you can always reach out to us at Duchenne@REGENXBIO.com, and you'll always get updates on clinicaltrials.gov on our listing here below. Beyond this, Rachel, sorry, I think we have one other plug here. If you are interested in being in our study, we do have the Affinity Beyond study. So this is a separate study from Affinity Duchenne, where we have a central primary investigator. That's Dr. Han Fan. She's out of Atlanta. She'll do a video assessment with you. And then if you're interested in getting tested for AAV8 antibodies to see if you could potentially be involved in our study, we will actually have a nurse come out to your site and draw to your house, wherever you want, come out to your house, draw the blood, and then we'll get she'll be in touch with you about whether or not you're positive or negative, and then obviously she can then refer you to any site that might be able to discuss eligibility for the trial at the different sites. So with that, thank you so much, and I'll go to your Q&A session, Rachel. Thank you.
Awesome. Thank you so much, Naz. I think that that was a ton of information. It was super helpful. I know that I have some questions. I see that we're getting lots of questions in the chat. So let's dig into it. I think a really easy one that I'm just going to quickly go to because it was the very last slide. So that Affinity Duchenne through Dr. Hanfan's site in Atlanta. So they would have to, if a family were interested in participating, they would have to go to Atlanta to do some baseline assessments, or it would all be done remotely
It's
at home.
It's all remote. So you would just
Okay.
do a video assessment where she would just ask you some questions about your eligibility for entry into the study, and then they would have a nurse. So we tried to make it as easy as possible. You could actually go to
Awesome.
your site if you live local and you wanted to do that, but everything is meant to be virtual and make it easier on the families.
Fantastic. And how many individuals are you recruiting for that study? Are you allowed to share?
I think we're looking for up to 200. I think that's probably on the clinical.
Up to 200.
Yeah.
Okay. All right. So I think
obviously
that one of
extend it
the
if we need to. It's fine.
Okay. I think, the reason I ask is because we think about commercially available products in the space. We think about other clinical trials in this space, and everybody's using a slightly different construct. Everybody's using a slightly different serotype of AAV. So maybe we could just start with you shedding a little bit more detail into AAV8. How is it different from AAV9 or AAV-RH74? How is it similar? What about cross-reactivity? I think that parents might say, well, we screened my child for treatment with another drug and he had antibodies, but how does that translate into a different serotype with a different drug? Can you help us understand that concept a little bit better?
Yeah, sure. I mean, I think I can answer, but since we have the gift of Olivier here, I'll let him do the talking.
Okay, but I'm sure you can answer, Naz. Yeah, thank you for the question. So we're using AAV8, and AAV8 is, the different AAVs belong to different families, right? They have close cousins, or they're more they're further apart from others, depending on the way they evolve. AAV8 is in one of these families that we call clades, and in this family is also RH74. So those are kind of cousins. They're not brothers and sisters. They don't look alike totally, but they have relationships, meaning that if you have an immunity that recognizes RH74, you may have some immunity recognizing AAV8 as well. That's kind of common. But there are also individuals who are not in this case. You could recognize one and not the other. It's possible, and that's why we want to be tested. So it's possible. AV9 now is further apart from those two. It's a different family, and it has different properties. It also has, I have to say, a different safety profile. There are things that are unique to high-dose AAV9 that we don't see in the clinic with AAV8, or that we that haven't been reported either with 74. So it has a slightly different behavior, and again, it's further apart. So the overlap, the chances that if you're positive for 9, that you're also positive for the others are less, but they're not null. I mean, I have to insist on that, right? I would say that it's probably 10% chances that you're not going to be that you're not going to cross react between those. So that's the situation. Again, those serotypes from different families have overlapping patterns. I mean, they are they look alike. They're all AAVs, and the immune system will recognize all of them together in a way, right? So that it's overlapping. Now we've chosen it because we have, you know, we have great expertise in the clinic with 8. We have great expertise in manufacturing 8 at very high purity. We know it's overperforming in our hands everything else with the muscle. So this is something that so far has been a great choice, and we're very pleased to see what we see in the clinic in terms of safety profile.
I think that's great and really helpful, Olivier. Thank you for providing that additional color around serotypes. I think it can get really confusing when there's so many different kinds. And I am seeing some more questions in the chat about it. So I want to clarify, and I think I know the answer, but I'm going to throw it to you all to clarify. So if somebody were to participate in this AAV8 screening study and they were negative, would there be potentially a pathway for enrollment in the treatment study?
Absolutely.
Yeah, for sure.
That's the point. I mean, that's one point of the study is to, you know how is the positivity in the population of Duchenne patients, but it's also it becomes also a source of potential patients. So these patients will have the option. It's not the only criteria for being selected, right? You also have the patient. You have, I mean, all kinds of things, right? That all kinds of boxes you have to tick, but this is an important one.
Very good. So what I'm hearing then is that it's really two-pronged. One benefit of this study is understanding things like prevalence of AAV8 in the general population, but also identifying potential participants for the clinical trial.
Right, exactly.
Great. Thank you so much. So I think my next question then is a little bit more about inclusion and exclusion criteria. I think, you know, in the community, we think a lot about where's your variant, what exons are affected, and how does that translate to potential treatment with a gene therapy. And all the trials are a little bit different, which makes sense to a certain degree. So I'm seeing some questions about exons 8 and 9, 1 to 17, deletion of 45. Can you just review again inclusion and exclusion related to variants within the DMD gene?
Yeah, sure. So, so we accept right now at present anybody with deletions or duplications in exons 18 and higher. We do have plans to expand. So like I said, keep your eyes peeled. Hopefully, like, you know, February, March next year for an expansion of that. We're working on that. And in terms of exon 45 deletion, we do not exclude exon 45 deletion patients from our study.
Okay. That's great. I think that that's really helpful. So that actually takes me to another question. And I feel bad because I'm asking my own questions and not necessarily questions that are in the chat. But talk to me a little bit then about how you're defining Duchenne, because I think, you know, there are certainly variants in the gene that can look like Duchenne and Becker, or maybe somewhere kind of in the middle. And if there aren't necessarily specific variants outside of that 1 to 17 that are being excluded, how are you really defining Duchenne as it relates to inclusion and participation in the trial?
Sure, it's a great question. So in the study, we actually leave it to the discretion of the primary investigator at the individual site to determine whether or not the child has a diagnosis of Duchenne. Honestly, if you get into the philosophical question of it, I see Duchenne and Becker in the spectrum of all dystrophinopathies, right? So it's a spectrum. It's really hard to draw a line between who is Duchenne and who is Becker. But that being said, to make life easier for all, we leave it to the discretion of the PI to define the child as having Duchenne. We do, so for the time to stand, though, we do have a lower limit of 3 seconds. So you can't be faster than that to enter into the study. So that would exclude some folks that are really high functioning. At this time, we don't have a designated upper limit to the NSAA that would probably, be something that we could consider in the future if we start seeing some patients that really don't fit the Duchenne phenotype, even though the PIs are calling them that. But in general, we have chosen very experienced primary investigators who are pretty good at defining Duchenne as any child who has, proximal weakness in the lower extremities and upper extremities, high CKs, it's beyond. I mean, I think we as clinicians even sometimes get a little confused about who's Duchenne Becker. Honestly, like when you're either severe Becker or an intermediate Duchenne, right? There's like a fuzzy
Sure.
point. But in my opinion, philosophically, I think it's fair that if someone has a deficit, a functional deficit, why not provide them benefit? So you can get into a philosophical argument about who might be able to enter into our study based on certain mutations, like if you predict in-frame would be, but we don't exclude in-frame mutations either if the PI says that this child looks consistent with the diagnosis of Duchenne. So I think to kind of get us all, because there are some in-frame mutations, particularly in the early region of the exons, that look like Duchenne. So you don't want to exclude people who could actually benefit from the drug. There are some patients who are exon 45 deletions who look like severe Duchenne as well, but some that perform better. So
Mm-hmm.
we don't want to close that opportunity to them. So we're just leaving it at the discretion of the PI to define the Duchenne diagnosis.
Okay. Excellent. Cool. That makes sense. All right. So then that makes me think about other sort of groups of people with Duchenne who would be interested in participating in a clinical trial with this drug. And I'm thinking about those who are adults or non-ambulatory. Are you able to shed any light on plans to include rather a broader group of individual participants?
Yes, we're definitely thinking about it. So obviously stepwise progression is our goal. Like, we have to kind of overcome some hurdles, but as you saw, we opened the study up to 1 to 3 year olds. We have plans to expand our mutation inclusion, and we have even grander plans in the future, hopefully, to expand even further. Because, obviously I've seen some of the folks chatting on here. Some of these are families that have approached me at meetings. They're tremendous advocates for their children. I see your face. I know who you are. I care about you. We care about you as a company. We care about your children. And we definitely want to be able to bring our drug to your child as soon as possible. So we're working on it.
Very good. I think it must be so challenging to hear soon, but not know when. And so we're grateful for your commitment. On the other side of the spectrum, you are unique in that you're including much younger kiddos in your trial, including 1, 2, 3-year-olds. Are you going to follow those kids any longer because of differences in sort of timeline and the point at which you would be administering drug and then being able to sort of see progression? What does that look like if I have maybe, an 18-month-old and I'm interested in participating?
Sure. So any gene therapy study right now is somewhat obligated based on the FDA to follow. Any child receiving gene therapy should be followed for at least 5 years after dosing. So we, in our phase 1/2 study, as part of the study, have 12-month data point. But once you're in the pivotal study, it's actually a 24-month data point for our functional data. But that being said, after that 24-month, you basically enter the same study, but it's labeled as a long-term follow-up. It doesn't have as many frequent assessments. It's like every 6 months of a check-in. But with those assessments are functional data that we're continuing to acquire. So we're hoping that a child who comes in at 18 months, for example, we'll get them at least until they're about four, about 4.5 or 5.5 years of age, for example, 6.5, and we'll be able to kind of see, how they're doing. We'll get that whole trajectory, and obviously, in the 4 to 6-year-old age group, you may see some improvement over time, but we're hoping that the SV95C that we're using to track activity will capture some subtleties that we might miss with traditional functional outcome measures. And then we've also uniquely instituted in that younger age group using the Peabody developmental motor scale to try to capture some of these changes as well. And then we're going to be comparing these patients during this phase, mostly on their rate of skill acquisition compared to unaffected children. So it's not going to be compared to there's no real natural history cohort for the Peabody in kids with Duchenne that are younger. So it's going to be compared to like rate of skill acquisition and how they're doing over time. We're trying to look at it from a unique angle to prove that it's not only safe, but also effective.
Excellent. Okay, I think that makes sense. That's really helpful. I do have a few questions in the chat about participation for folks living outside of the US. And I think we've seen a few different models in the community for international participation. Does REGENXBIO have any insight into the ability of folks from outside of the US to participate?
Yep. So we are definitely open to having folks come out from outside the US. Obviously, there's some limitations that come with that. You need to have a visa. You need to make sure you have some insurance in case, God forbid, something happens while you're here, that you're covered. These are all institutional requirements as well, like for the different sites that are running the study. And then obviously, we just need a commitment from the families that you'd be willing to come back for all the follow-up studies, because it would be very detrimental to the study if you came in just for the treatment and then just never came back. So, but with that commitment, if if you're interested and you're from outside the US, please contact us at Duchenne@REGENXBIO.com, and we'll direct you to the different sites that may be able to accommodate you. And once you qualify for the study, obviously you would be supported with your travel and whatnot to be a part of the study moving forward.
All right. So I want to go back to some of the data that was presented. And I don't know if it would make sense to pull that slide up, or maybe we can just talk about it. But you were talking about improvement in some of the different outcome measures that were used, like the North Star, like the time to rise. And I think for me, I got a little bit confused. And so I'm hoping that you can just clarify for me. When we're saying improvement, are we saying improvement for that individual? So I was at 10 seconds before and now I'm at 8 seconds, or are we saying improvement compared to sort of these age match controls in natural history? Can you just clarify that piece?
Yeah, sure. I mean, there's a lot of data here that we had to summarize in these figures. For the dose level 1, since this is not our selected dose, we didn't put the individual data points in for the patients. So this is an average of all the patients. And what you see here in the gray box is what you would expect to be the trajectory in the time to stand over 1 year for this average age of this group. And then here is what you have as an average improvement. And so we added the -1 to like the point, the 2 here, and we added it to get the delta of -2.9 compared to natural history. But there's still an improvement of about 1.1 seconds on average from baseline. Does that make sense? So you're improving.
It does.
So, but here on the dose level 2, we actually do present the individual data points for the patients. So because we only have the 2 patients, but just remember that this is 9 months after dosing. So here at 9 months on this 8-year-old, he was stable on his NSAA, not worsening, but you would expect at 9 months to be a little bit, I'm sorry, on his time to stand, you would expect him to be a little bit worse on his time to stand 9 months later for this 8-year-old. Because again, this is an older patient, right? And with his baseline characteristics, you would expect his trajectory to be like at 9 months to slow down by about 1 second. And he improved. This is a -0.9 second. In the 10-meter walk run, he improved here like by about, I don't have the actual numbers listed here on the bars, but it's like, I would say, don't quote me on this, by about 3.3 seconds he improved. But then, here compared to what you would expect in 9 months for an 8-year-old with his level of function at baseline, he should have declined by about 0.5 seconds. And the same with his time to climb. So his time to climb, here you would actually, the natural history was such that there would have been a little bit of an improvement. So we had to subtract that from the actual improvement that we saw from baseline. And then we got an improvement compared to natural history of 1.2 seconds, but it's about 1.1 seconds. And that's the same for the 12-year-old and the NSAA as well. So what you see, we didn't actually have an external natural history control for the 12-year-old, and his baseline, the comparison between his baseline NSAA and his 9-month NSAA is that he went up 10 points The 12-year-old went up 10 points.
So maybe we can talk a little bit more about that, because
Mhm.
I think a lot of folks who are listening in right now see that and say 12-year-old,
Mhm.
10-point improvement on the NSAA, like that's going to stop a lot of folks in their tracks, and they're going to be like, whoa,
Yeah.
let's talk some more about what this means, because that's really exciting. Can you shed any more light on what that child looked like at baseline and clinically? Like, would we have expected this type of response, or do we have any hypotheses around why we have seen such a robust response? Is there anything about his genetics that would provide additional context here? What can you share?
I could talk a little bit about his baseline, and then maybe Olivier, if you want to comment on what we would have expected for him, since you're here as well. But basically, his baseline, again, defined by the primary investigator as having Duchenne. We do have a video of him at baseline that we can compare to his follow-up, but unfortunately, I think because it gets posted on your website, we can't just leave the video up on the website, or there were some issues with showing the video. But on his video, he definitely has proximal muscle weakness. He had difficulty going up the stairs at baseline, where he was pulling himself up with his hands. Obviously, any 12-year-old entering into our study by default is going to be more of an intermediate phenotype of Duchenne, right? Because you have some kids who lose ambulation at around 10 to 12 years of age. And then you have some kids that lose ambulation around 18 to 20, right? So he might be, and you're still losing ambulation, but you might just have a little bit of extra time of ambulation. So he might be somewhere at the later end, possibly, or maybe somewhere in the middle between these two. We don't know what his exact phenotype would be without intervention. But what we do know is that he had proximal muscle weakness at baseline. He had a positive Gower sign. He had difficulty going up the stairs. This is like very clear in the videos. He didn't have a clear run. He was dragging his toes when he was running. And then 9 months after our intervention, he's going up the stairs with a lot more stability. He's not having to pull himself up with his hands. He's getting up off the ground a lot quicker. And then he's able to hop. And he's able to clear his feet and go into a clear run. And this is a child that previously it might have been at the PPMD meeting that we showed his video, where the parents got very excited because he was staying at the trial site and was riding his bike in the hallway at the hotel. And the parents said he's never ridden a bike before. In the video that we showed, he rode his bike 6 miles after at 9 months. So he is doing remarkably well. We're obviously, I'll defer to Olivier now to kind of talk about why we think he's doing so well. Yeah, no, we obviously, but this is N of two patients, right? We need more time also to see how things evolve, and then we'll take it from there. So Olivier, I'll let you chime in as well.
Yeah, so again, we're really excited as everyone about this patient, but we want to remain careful. It's one observation, one patient. What's absolutely clear to us is that it's not that, you know, there was a mistake at diagnosis and he was actually doing very well and we treated him and he's still doing very well. There is a huge differential between his performance at baseline and what he's doing today. So we did something to this patient, right? Now, why is he such a good responder? Is the question, right? Why is he responding in such a spectacular way? Maybe by being a intermediate mild Duchenne or severe Becker, he's in a category of patients which are primed for good response, right? And we'd like to understand this more. Is it genetic? Is it something else? The jury is still out. This patient has, it often happens. But this patient actually has some low level of baseline signal that reacts with the dystrophin antibody. So he must have some kind of in-frame dystrophin being produced. It's very low. It's a few %. And as far as we can tell, still need to be analyzed. In this region where he has a mutation, there are possibilities for putting back by some natural exon skipping happening in the cell, the frame together. But so far, everything which has been characterized in terms of putting back these frames together, it creates proteins that you can detect, but they're non-functional. They're always associated with Duchenne, right? Now, maybe they have something. Maybe these proteins kind of, in a way that we don't understand, help our microdystrophin to work even better. That's what we're trying to figure out. But so this is all work that we're doing. I can't, tell you today what's going on, and we want to get to the bottom of it. But again, I repeat that what we are doing something very clearly to this patient and in a very spectacular way. And what we're excited about is that we may be able to, by understanding that, define a group of super responder patients, right? And if you want to call them mild Duchenne or severe Becker to start with, I mean, doesn't really matter, right? What's clear is that this kid was, as a kid, very early on diagnosed as Duchenne. He's not a Becker of the type you discover when the patient is 40-year-old and it's diagnosed by chance, right? And it's been normal up to there. He's a severely sick kid, but the great thing is that he's responding in a spectacular way. So we're looking forward to understanding more about that, definitely.
So I think that's really helpful. And I think, I just look at it in the larger landscape of like response to drug. And it sounds like, this kid's doing really, really well, which is really exciting, but it also highlights some of the questions that we have as a scientific community trying to understand whether it's genetic modifiers or whatever this, you use the term super responder, how that works, what it means. And how can we leverage that for the larger community too? Like, is there something to this? So I really appreciate you diving in a little bit deeper, just because this does seem like such a significant response, and I think it's helpful to understand. I want to recognize that we're at 3 minutes until the end of the hour, and I still have gobs of questions, but I'm going to have to shorten my list up and hit the highlights. Maybe you could clarify for me, Naz, as we think about regulatory terminology, you talked about pivotal and you talked about phase 3 and what this means for, I think I heard a 2026 potential submission to the FDA. Just quickly refresh me, what are the differences between pivotal versus phase 3, or can they be used interchangeably? And then what does this forward-looking timeline look like for the community who's really excited?
Yeah, it's a great question. So phase 3 is just, we've kind of established the safety profile for our drug, and we're checking for efficacy, right? So our pivotal cohort of 30 patients is mainly what we're aiming to use for accelerated approval, hopefully, so that we can kind of get our drug out there after we dose our 30 patients if the FDA agrees with the data that we share at that point, that our microdystrophin levels are good, greater than 10%, have at least about 50% have at least greater than 10% expression of the microdystrophin, showing that our particular microdystrophin, so not comparing, because it's really hard to compare between different companies, but our particular dystrophin is getting to the correct place and is expressing itself correctly, and we think it's likely to predict clinical benefit. But obviously, we're also going to show some functional data to support that at that time when we have our 30 patients biopsies done and microdystrophin levels. So that's the pivotal portion of the phase 3 study. And then the confirmatory portion is probably going to be more fixated on the functional rather than the microdystrophin.
Okay. And then very quickly remind me, what are the primary outcome measures?
Sure. So the primary outcome measures are a little different between the 1 to 3-year-olds and the 4+-year-olds for function. So everybody right now is the primary endpoint, as it stands right now for the pivotal portion of the study, is the proportion of patients with microdystrophin levels that are greater than 10% across the board. So we aim to have everybody express it at greater than 10%. But the secondary endpoints for the 4 and older group is going to be the timed function test, so both in seconds and in velocity, and also the SV95C. I'm sorry, not as a secondary endpoint for the 4 and older. So it'll be the time function tests primarily, and then for the exploratory outcomes, it'll be the NSAA and the SV95C for the older patients. For the younger patients, the secondary endpoints are going to be the SV95C and the Peabody motor developmental scale.
Excellent. This was a ton of information. Naz, Olivia, thank you so much. I think this has been really helpful, really insightful. I think if I had to very briefly summarize, it sounds like if families are interested in potentially participating or learning more, they could reach out to that Duchenne@REGENXBIO.com address, or they could probably reach out to the centers that are dosing sites for your clinical trial. If they're interested about antibody screening as a potential mechanism to participation in the trial, or just being citizen scientists, they could reach out to Dr. Han Fan at Rare Disease Research in Atlanta to talk about AAV8 screening. I think I heard that there's a really exciting timeline ahead of us with this pivotal study with a potential 2026 submission. Obviously, timelines can change and the regulatory process is long, but this is still really encouraging, and that we've seen some improvement in some of these kids compared to natural history cohorts, and that perhaps there are some other questions that we've identified as a community to help us understand what these super responders are doing so that we can make use of that and expand participation to as many folks as possible, young, old, and everything in between. Naz, I want to give you the last word before we wrap. Is there anything else that you want to leave the community with today before we finish up?
Sure. I just want to say that we're super excited by the potential of RGX-202 based on what we're seeing. We anticipate and have confidence that we're going to perform even better across the board. You know, we encourage all of you, if you're interested in participating in the study, to reach out to us. We have a great safety profile. You'll be in good hands with the sites and us at a distance. We encourage you to be part of history in the making, which is basically the field of Duchenne gene therapy at this point. So we're excited to be able to provide this therapy, and obviously we're open to any discussion or questions that you have at any time.
Great. Thanks so much, Naz and Olivier. As a reminder to everyone, we're really grateful for you joining. This is recorded. It will be available on our website in the next few days. And if you have questions for the PPMD team, you can always reach out to us too. You can shoot me a message directly, Rachel@parentprojectmd.org. And then you can reach out to the REGENXBIO team, and we'll include all of this in a quick recap that will go out to the community along with that recording. And with that, we're going to go ahead and conclude today's webinar. Thanks again, everybody, for joining us. We're grateful for you.
Thank you so much. Thank you. It was a great pleasure to be here.
Bye-bye.
Bye.